How do I get it?
Oh,
There we go.
Ted's got it covered.
Okay, so I'm Stephanie Arsenault. I'm part of Yang Chen's lab here at Stony Brook. Also be going over kind of not a traditional AFS type of talk, but something that I think is super important. So first, let me just talk a little bit about the Hudson River Biological Monitoring Program. It was a monitoring program of the Hudson River from Battery Park up to the Troy Dam in Albany. It had different survey components over the course of the survey period. The Long River Survey, Fall Juvenile, and Beach Seine, so the ones that are underlined, provided data on the abundance, stage structure, and spatial temporal distributions of fish species in the Hudson. There were some other survey components, such as a sturgeon tagging program, some stock assessment programs, and the impingement and entrainment programs. And so this was conducted from 1974 to 2017 with different surveys occurring at different time periods. And the database and the biological samples collected during the survey were donated to Stony Brook by Entergy. So moving on to metadata, what is it and why do we need it? So metadata broadly is the data about the data. It should provide documentation of all the variables that were collected. It needs to provide the necessary background information, all of the data processing steps that were done, and it should be comprehensive, informative, and easy to use. And so all of these components are to facilitate the end user to use the data appropriately in a manner that is most suited to the data being provided. So why do we specifically need data for the HRBMP? It's a huge database. So we have data from 1974 to 2017, multiple different survey components. It's a pretty complex database at the moment. There have been changes in survey protocols, and there are some data use caveats associated with the compiled data. And all of that would affect the data that the end user receives and how they should be using that data. And just how it is right now, it's difficult for the common user to understand. It's taken a lot for our lab to kind of piece through some of this stuff. And so metadata is one tool to help address those complications. Just to highlight some of the types of data that was collected from a single survey, the Long River Survey, which is an ichthyoplankton survey. This is not a comprehensive list. This is a fraction of the variables that were collected. So we have temporal and geographic information. We have stuff such as vessel code, tow speed, so descriptors of how the gear was deployed and how the data was collected. And so in a metadata record, each variable needs to be included, it needs to be defined, it needs to have an appropriate explanation and documentation. That way an end user, looking at a column of data in a CSV file or in a database understands exactly what that data is showing. And so another way of doing this is a lot of times you can be provided with a data dictionary, which just has, like keywords, variable names, and then definitions. Just to further highlight the complexity of the Long River Survey data, so these are some of the survey protocol changes that occurred over time. So like I said, the survey ran from 1974 to 2017, and some changes were made. So for example, the species of interest changed over time. So some species were added and removed. The day or night sampling, when it started or ended each year, varied throughout the survey. The number of river runs and the number of samples collected may have differed based on when the survey started, if there was any inclement weather, if they had to change their sampling plan for any reason. Another complicating factor is that over the course of the entire survey period, different contractors performed the survey. So we had like Texas Instruments, Norman Dow, ASA, and so different people in the industry were conducting the sampling, which does affect the data that the end user is provided with. And then another example is that the allocation of sampling effort within each region and strata differed. So sometimes it was based on a stratified random sampling, other times it was based on the distribution of fish observed in the previous year. So all of these types of information need to be provided and explained when you're giving data to an end user for something that's so complicated. So we came up with this 6-step method, I split it up into phase 1 and phase 2. So phase 1 is the top 3 steps. In blue, it's more of the development phase, so we're identifying our metadata reviewers, we're creating a data subset, and then we're going to develop these metadata prototypes that will be sent to the reviewers. And then phase 2, in green on the bottom, is more of this consulting and refinement phase, ending in developing the final metadata products. So I'm going to walk through a little bit more in detail, like what each step entails. So starting with phase 1, this kind of introductory phase. So we started by identifying our metadata reviewers. This simple pie chart is just showing where the metadata reviewers are affiliated. So we've identified 20 reviewers as of right now from different sectors, including the industry, state and federal agencies, academia, and nonprofits. So we have people coming from the Hudson River Foundation, New York DEC, Normandow, UConn, Cornell, and more. And so we wanted to select people who are familiar with the HRBMP data, people who work in and around the Hudson River estuary, and we did make sure to select some people who are outside of the area to provide a more outsider perspective. The next step is subsetting the HRBMP data, so only working with a small fraction of data to develop these metadata prototypes. So I'm working with data from a single survey, the Lund River survey, with a single species, very charismatic bay anchovy, and I'm only using egg data. I am only using data that was collected at night, using a tugger trawl, and I even pared down the years from 1982 to 2017, using only months May through August. And so this data subset is one I'm working with on another project, looking at bay anchovy suitable spawning habitat, and I wanted to work with the same data subset, because that's what I'm most familiar with, and not creating any additional unnecessary extra effort for myself at the moment. And so to keep it even simpler, I have pared down the number of variables that are going to be included in the metadata. So I only have 9 that a metadata reviewer will be provided with. So it's enough temporal and geographic information to go along with the egg count to provide more of like the context. And throughout this entire process, I'm also keeping detailed records of any processing steps and data manipulation that was done. That way the end user knows exactly how the data that they get sent was developed. So now that I have this data subset, I'm going to develop these metadata prototypes, and so I have 5 different ones that I'm going to go through. The first 2, the FGDC and the ISO, these are 2 metadata standards that were developed within the US, the FGDC, and then the ISO is an international metadata standard. And both of these are used by federal agencies such as the USGS and NOAA Fisheries. And even though the standards themselves differ a little bit, the end product is the same. They require that metadata is formatted in extensible markup language, or XML, and there are a couple of free tools to create and edit XML metadata. The third is kind of moving in a separate direction. It's based on EMAP, which is the monitoring and assessment program. And so the way they provide their metadata, it's more on a species-specific level. So the user will be provided with a CSV with a general write-up of the data being provided, some summary figures and tables. And then the 4th and 5th are just combinations of the metadata standard with the EMAP model. And along with each of those 5 models, the metadata reviewer is going to receive a description of the Long River survey and any protocols that were included, or any protocol changes that occurred over the survey period. So kind of some of the stuff that was highlighted in that previous table, just to provide as much background information as possible. So this is just a screen grab of the Metadata Wizard. It's a desktop application developed by the USGS that I'm using to generate the FGDC metadata standards. Its icon is this really cute rubber ducky. And one of the nice things about this application is that it can auto-populate data. So if you're providing a CSV file that has like 40 columns of data, it can auto-populate that information, saving you a lot of time. So this is an example of what FGDC metadata in XML format looks like. I know it's kind of clunky, but it always starts out with some origin, when it was published, abstract, purpose, and another way of displaying the same information it's still an FGDC metadata record, it's just displayed with HTML format instead. So you could provide this to reviewers instead of like the clunky XML, even though it's the same information. I don't have an example of the ISO metadata at the moment, so I'll just go into the NYCAFS metadata example. So like I said before, the data is provided in CSV format. There is no XML metadata. There are summary figures and tables, so like a paragraph describing the data, and some figures and tables that are in the same CSV file, just on like a different tab, just providing some very brief explanation of the data being provided. And so I grabbed this figure from the 2016 HRBMP year cost report. It's just showing temporal and geographic indices of bay anchovy eggs from 1988 to 2015. And I included it just to highlight, like, this is a type of, summary figure that you could develop and include in like a NYCAFS metadata model. So moving on to phase 2, the more like refinement step, so the consultation with stakeholders, getting their feedback, selecting the final metadata, and then developing the final metadata. So we're going to consult with the stakeholders that we've identified through different surveys, letters, seminars, or status reports. Right now we have a survey drafted, and we're going to work on some other ways to interact with the metadata reviewers through incorporating their feedback and a discussion within the HRBMP research group. We're going to pick the metadata model that is most appropriate to be used moving forward. And then we're also going to ensure that the stakeholder feedback is incorporated into the final metadata. So we're going to ask the metadata reviewers like what you liked best about this prototype, what didn't work for you, what type of information should be included, did we miss anything, was anything overkill, that type of stuff. That way we are keeping in mind the end users when we're coming up with the final metadata. And then finally, we're going to develop the final metadata. So starting with the Long River survey, so we're going to finalize the metadata for the Long River survey. So expanding from that like little data subset, including all data collected by the Long River survey. We're then going to develop metadata for other surveys within the HRBMP. So we have the beach stain survey, fall juvenile, and water quality survey. Additionally, we're also going to be making sure we have a completed documentation of any survey protocol changes. So kind of coming up with a separate standalone PDF explaining the history of the survey and what changes that occurred. That way we can provide the end user with additional context information. We're also going to generate an online publicly accessible metadata. So we want the metadata to be published, accessible to all end users so they can just take the first step of trying to figure out what type of data we have. And then we're also going to finalize a data sharing policy. So in the future, people are going to be able to make data requests. We will process them and then provide the person with the data that they're requesting and then the appropriate metadata records. And with that, I would like to acknowledge some of the important contributors. So the Hudson River Foundation, Entergy, New York DEC, SOMOS, and then the HRBMP Research Group. And if you want to learn more information about the HRBMP, its history, or keep up with the exciting research opportunities our group is doing, you can visit our website.